Welcome to AP Biology. This video is on the Meiosis Lab. The goal of the Meiosis Lab is to determine the frequency of crossing over and therefore the distance between two genes. In order to do this lab, we're going to use an organism called Sordaria, and Sordaria is a type of fungus. We know that fungi are haploid organisms, and since they are haploid, they do not need to mate with each other in order to reproduce. Instead, they could reproduce asexually by producing these structures called spores. When these spores are released, they're released into the air, they land somewhere, and wherever they land, they can simply grow through mitosis into a new Sordaria fungus. The spores that the Sordaria produces are contained in the structure that you see right here called an ascus. So here you see the ascus, and here you see all the spores inside of it. The problem with the Sordaria reproducing asexually is that asexual reproduction does not produce any genetic variation. So when two Sordaria come into contact, you see right here, they will mate with each other. They fuse together and they create this diploid zygote, and that diploid zygote immediately undergoes meiosis, returning it back to being haploid. Well, we know that during the process of meiosis, genetic variation is created, and that's going to allow the Sordaria fungus to evolve over time. One of the major methods by which genetic variation is created through meiosis is crossing over. Now, in this lab, we take two different types of Sordaria. One type of Sordaria, which is the wild type, the natural type, is black, and the black Sordaria produces black spores. So we could pretend that this one right here is the black Sordaria. The other Sordaria is a mutant. It is tan, and it produces tan spores, and we could pretend that this one right here is the tan Sordaria. When the two fuse together and they create the diploid zygote immediately undergoes meiosis, returning it back to being haploid. If you look at the arrangement of the spores inside the ascus, and this is the key part, if you look at the arrangement of spores inside the ascus, you can tell whether or not crossing over took place between the two Sordaria. If no crossing over took place, then you're going to have the simple alignment of 4 tan and 4 black spores inside each ascus. It could be tan then black, or black then tan. If you have any arrangement of 2, 2, 2, and 2, this showed that when this diploid zygote went through meiosis, that crossing over took place, and it rearranged all of the spores inside of the ascus. Now, your goal for this lab is to look at a picture that I am going to hand out to you, and you're going to calculate the frequency of crossing over. This is going to be the picture that you're going to be given, or you could use the picture on the board if you would like, and you're going to count the total number of asci. You're then going to count the total number that show crossing over taking place. So anytime, like this one right here, where you see a 2, 2, 2, 2 arrangement, this is an ascus that shows that crossing over took place. You're going to take the total showing crossing over, and you're going to divide that by the total number of asci, multiply that number by 100, and that's going to give you the percent crossing over. We know that the percent crossing over, or the frequency of crossing over, is directly proportional to the distance between two genes. When two genes are further apart, the frequency of crossing over is going to increase. When two genes are close together, the frequency of crossing over is going to decrease. we could use the percent crossing over to figure out the map distance between two genes. To determine the map distance, you're going to take the percent of crossing over and you're going to divide that number by 2. So if 50% of the ASCII show crossing over, then those two genes, whatever those two genes might be, are 25 map units apart. Use the worksheet that I give you to calculate the percent crossing over, and use the data table on the back to figure out the map distance between those two genes. This concludes this video.